morning. Today we are going to talk about mineral metabolism, the series of seminar. I'm going to start with the molecular basis of physiology of mineral metabolism. So what are minerals? Minerals are inorganic elements which are found in nature, mostly in the soil. From the soil it goes to the water, and the water plants absorb the minerals. Through plants we are able to, and animals we get minerals inside our body. So, is our body able to produce our own minerals? The answer is no. So, we get minerals only from the environment. And minerals, are the minerals stored in our body? Yes, some minerals are stored in our body, some are not stored in our body. What are the minerals which are stored in our body? It's calcium, phosphorus, iron, zinc, copper, magnesium, iodine and selenium. And the minerals which are not significantly stored in our body are sodium, potassium, chloride, chromium, fluoride and molybdenum. So, the picture is not seen. When we see the human body, 62% is of water and 16% is protein, another 16% is fat, and the remaining 6% will be minerals, carbohydrates and other substances. So minerals occupy very less portion of the human composition, but it plays very important roles in the bodily physiological function. So what is mineral metabolism then? Metabolism refers to how the body uses, stores and regulates the minerals. Regulation in the sense when there is high mineral, it has to control the absorption. When there is low minerals, it has to increase the absorption. So that is metabolism in basic. So mineral metabolism is how the body controls the minerals in our body. Today I am going to talk about calcium and phosphorus metabolism mainly. So physiology of calcium ion and PI. PI stands for inorganic phosphate. Why I am telling us this as ion and calcium as ion and phosphorus as inorganic? Because phosphorus, the elemental form, is toxic to the human body and it is always absorbed and stored in our body in the form of phosphate. So we always refer that as the inorganic phosphate. So it is in the form of H2PO4 or HbO4 to the power minus. So this is the reason I am mentioning it as Pi. Calcium will be mentioned as Ca2+. It is an ion. So tight control of plasma, calcium and phosphate levels is essential for many vital physiological functions. Several organs contribute to the regulation of calcium and phosphates homeostasis by facilitating intestinal absorption, bone mineralization or demineralization, and renal excretion or reabsorption of both ions. So what are all the organs which are playing a major role in the metabolism of minerals is, first it has to get absorbed from the intestine, and then it has to go from the blood it goes to the kidney, where it gets reabsorbed or excreted, and from there it enters the bone and other areas of the body. So the hormones also play an important role in regulation of these processes. Biologically active form of vitamin D, which is 1,25-dihydroxyvitamin, which is D3, parathyroid hormone and calcitonin. These are the hormones which are playing an important role in mineral metabolism. And there are some growth factors like fibroblast growth factor number 23, FGF23 and TOTO. These two have been identified as the new players in essential for regulation of calcium and phosphate homeostasis. So we will be seeing about this FGF23 and TOTO throughout the mineral metabolism seminars. So the dietary phosphates and calciums intake produce only small alterations circulating levels of these ions due to the combined action of these signaling molecules on bone, intestinal and renal calcium and phosphate transport. So when we just see this is the fibroblast growth factor FGF23 and this is the cloth of protein. So they either act together or it's FGF23 alone will act. This will help in this is the molecular level of mineral absorption, which I will be talking in detail in the next section. So first the calcium and phosphate transport protein. So once we intake the minerals through our food, I said it is only through food we take the minerals. So when the calcium and phosphate ions enter the intestine, what happens? The small intestine only plays an important role in the reabsorption of the minerals. In the small intestine, we have different regions. The duodenum and the ileum plays an important role in the absorption of these minerals. So from the duodenum, the calcium gets absorbed, whereas from ileum, the phosphate ions get absorbed. So why only in those particular areas are they getting absorbed? It is because these areas have the members to take the calcium or the phosphate ions inside the intestine. So active intestinal calcium absorption begins with the luminal entry mediated by the epithelial calcium channel and TRP basis. So these two are the main receptors of calcium ions which is present in the duodenum. So calcium is subsequently shuttled to the basolateral side of the cell via calcium binding protein which is called as cal binding D9K. So when I show in the picture it will be clear. So if this is the cell, this is a cell of the intestinal lumen, or that is the duodenum. This is the basolateral and this is the lumen here. So we have the TRP B6 channel here through which the calcium enters, and this is the cal binding D9K which takes the calcium, brings to the basolateral side and then leaves it outside through this PMCA1B which is this plasma membrane calcium adenosine triphosphate. So through this, if there is one more channel, I will talk about this later. So this is the process of absorbing calcium from the duodenum into the blood. So from the lumen into the blood, this transport happens with the help of TRPV6, cal binding and PMCA1B. So the sodium dependent, now we will talk about phosphate. The sodium dependent PI cotransporter type 2B. So in short form we call it as NAPI2B is localized to the brush border of the ileum. As I said, calcium in the duodenum, phosphate from ileum. In the ileum we have this NAPI2B, and there it is responsible for active PI transport. PI is inorganic phosphate from the intestinal lumen into the blood. So the NAPI2B is also present in the duodenum, but in a very significant, very less amount. So the absorption of phosphate is very less in the duodenum. So the picture is not seen. That also the Na2PI takes the calcium with phosphate ions inside the lumen, takes it to the basolateral side, and it gives into the bloodstream. So next is, once it has entered the bloodstream, where does it go? It goes to the kidney. In the kidney, we have the smaller units, the basic units of our nephrons. So along the course of the nephron, passive paracellular calcium absorption occurs from the proximal convoluted tubule and the thickened ascending lumen. So I have a picture which is, this is the phosphate entry. This is through the NAPPA, and then it gets absorbed into the bloodstream. In the nephron, there is a three-step process which mediates the active calcium reabsorption mechanism from the distal convoluted tubule. We know kidney, there is a glomerulus, then there is a proximal convoluted tubule, then there is a descending and ascending limbs, and there is the distal convoluted tubule and the main connecting duct there. So that is how the nephron is, we have to just imagine, it is not shown. The three-step process occurs in the distal convoluted tubule. The first calcium enters the cell. So this is the picture of a nephron. You can see the Bowman's capsule, the proximal tubule and the descending and ascending limbs, and the distal convoluted tubule and the collecting ducts. So this is the nephron. Calcium absorption actively takes place in the distal convoluted tubule. That is this. So calcium enters the cell via epithelial calcium channel TRPV5. So in the nephron, the receptor is TRPV5, whereas in duodenum it was TRPV6. Then the calcium diffuses through the cytosol via calbinding D28K. Like we had a calbinding D9K in the duodenum, we have calbinding D28K in the nephron to the basolateral side of the cell, and there it is extruded into the peritubular capillary by the NCX1 and PMCA1. So this is how. We have to, when we discuss about the molecular mechanism, we see from the cellular point of view. So in the cell, in a particular cell, how the calcium enters and how it is taken to the basolateral area, and then how it is taken into the blood. This is how the reabsorption of calcium ions takes place in the nephron of the kidney. And how does the inorganic phosphate gets absorbed is by active absorption, which is also through the proximal convoluted tubule. So this happened, calcium absorption was in distal convoluted tubule. This is in the proximal convoluted tubule. This is the glomerulus, the Bowman's capsule, and here is the proximal convoluted tubule. And cellular level, if we say, we have all the receptors here which take and then transport it outside. So active phosphate absorption occurs in the PP of the kidney via the sodium dependent transporters. That is, there are two types, 2A and 2C, with high expression levels in the early segments of the proximal convoluted tubule. Now it has entered the bloodstream. From the kidney, it is reabsorbed. What are all the other regulating factors? We have the hormonal regulation of the transcellular calcium reabsorption. So what are all the hormones which are involved in this process? It is the parathyroid gland. So we know parathyroid gland is essential for the calcium homeostasis. In the parathyroid gland, how it happens? There are receptors called as calcium sensing receptors, CASR, which are responsible for sensing the levels of calcium in the blood. So when there is hypocalcemia, then it inhibits the CASR, which in turn promotes the secretion of parathyroid hormone. This hormone stimulates the calcium mobilization from the bone and the conversion of inactive vitamin D to active form with the help of the cytochrome P450 enzyme. And then the increased active vitamin increases the vitamin D receptors, which mediates the gene transportation, resulting in an increased transportation of calcium transport proteins, resulting in the stimulation of calcium absorption or reabsorption. So this is how the parathyroid hormone helps in case of hypocalcemia. And when there is hypercalcemia, this activates the CASR, thereby inhibiting the parathyroid hormone. And likewise, the calcitonin is released, which decreases the osteoclast-mediated bone resorption and promotes lowering the blood calcium concentration. So parathyroid hormone helps in either decreasing or increasing the levels of calcium depending upon the situation of the blood levels. What is the next hormone which plays an important role? It is the estrogen. So it contributes to calcium homeostasis by promoting bone mineralization and stimulating the renal TRP5 expression. So there it plays an important role, which results in increased active calcium reabsorption independent of vitamin D3. And estrogen exerts the effect on renal mediated calcium handling by altering at the transcriptional level, that is the expression of calcium transporters. So during when there is a low estrogen, that is the reason that there is more of calcium loss in women. And one more factor is tissue cadherin, which is called TK, which is a proteolytic enzyme that is secreted into the tubular fluid by renal epithelial cells, which activates the bradykinin receptor, stimulating the phospholipase C and the DAG-PKC pathway. So this TK increases the TRP5 mediated calcium transport. So we can see here, this is the TK, which is getting attached to the bradykinin receptor, and this is the pathway which I told. PLC-DAG pathway, just it happens here, and it stimulates the TRP5 receptors for the calcium reabsorption. And there is another hormone called as the anti-aging hormone, which is called clotho. Clotho is a type 1 transmembrane protein. It is a transmembrane protein. This is a cell membrane, but there is a protein attached, which is the clotho. With the beta-glucuronidase activity, and this activity is contained in the extracellular domain. So it happens in the extracellular region that can be secreted into the blood, urine, and cerebrospinal fluid. The stimulatory effect of clotho on TRP5 substantiates the important role of this anti-aging hormone in calcium homeostasis, specifically by altering the luminal membrane of calcium permeability of the DCT and the cortical collecting count. So this is the these are the regulators of hormonal regulators of calcium ions. What are the hormonal regulation which happens in the phosphate reabsorption or absorption? It is vitamin, parathyroid hormone, vitamin D3, and dietary phosphate intake are known to alter the phosphate absorption or reabsorption. Clotho and FGF23 were recently identified as new key players involved in the regulation. So an increase in intestinal sodium that is transporters expression can be stimulated by either vitamin D3 or low phosphate intake, ultimately resulting in elevated blood phosphate levels. PTH, which is parathyroid hormone, increases blood phosphate levels by inhibiting the reabsorption from the proximal tubules. the overall